I know Avi since he was a graduate student at Mount Sinai, and it has been a pleasure to see him grow and having a lot of impact in the area of computational biology in general. His tools are extremely well used, and you can probably, some of you, I'm sure, must have read papers that use his tools. What is the name? Avi is director of the Mount Sinai Center for Bioinformatics, professor in the Department of Pharmacological Sciences and the Department of Artificial Intelligence and Human Health, and a faculty member at the ICANN Genomics Institute. He is the principal investigator in this NIH Common Fund Data Resource Center, which he will tell us about, for the Common Fund data ecosystem. His lab develops widely used bioinformatics software applications for the analysis and interpretation of high-throughput omics datasets. Avi's research focuses on applying statistical learning techniques to understand how intracellular regulatory systems control cellular processes, including differentiation, dedifferentiation, apoptosis, and proliferation. So I think it's going to be a treat to hear how he's thinking of data in the context of the Common Fund data ecosystem. Avi, please connect with this.
Thank you. Thank you, Gustavo, for the invitation. It's a pleasure to be here today. Like Gustavo said, I'm representing the CFDE and also the CFDE Data Resource Center. That I'm contact PI. There's another PI, Shankar Subramanian, who's also very well known from UC San Diego. That he was also invited to speak today, but he's not available today, so I'm representing both of us. And I'm going to talk about the CFDE Workbench, which is a resource that we developed for this CFDE Common Fund program. So the CFDE Common Fund program was established to try to connect data sets from across Common Fund programs. This is very challenging. Those Common Fund programs, they are producing very different types of data. They're basically addressing different roles, but the idea is that there's a lot of opportunities for combining data sets from across Common Funds for additional discovery and also organizing those programs. So you guys just got started with complementary, and those programs also end in 10 years. So in 10 years, there is also this challenge of like, how do you maintain the resource after the mandatory ending of those programs? Because typically they're becoming very useful for the community. So we have to deal with that. So the NIH established this CFDE program, and it has 5 centers, and we are one of those centers. So we are the Data Resource Center. There's also the Knowledge Center, Training Center, Cloud, Workspace, and the Admin Centers, which is the Integration Coordination Center. So all of those centers, they're supposed to work together to produce this resource for the community. And on the right side, there is this wheel of the Common Fund programs. So all of the Common Fund programs that are in the wheel, they're participating in the CFDE. And I think it's mandatory for you guys to also participate in the CFDE. So there is some requirements that I'm going to talk about and how we are hoping to engage with you. So the vision of the CFDE Workbench, or the Data Resource Center, is that we're going to take data and metadata from all of those Common Fund programs, and then we will harmonize it at the metadata level and also at the process data level, and I'll talk about that. So there is a data submission system that we established where the DCCs, or the data coordination centers from all of those programs, they are preparing those metadata files that I'm going to discuss. They are in this harmonized metadata standard that's called C2M2. And then once the data is formatted, it's all metadata. So it's not really the actual data. It's the metadata that describes the files that actually have the data. And then once it's in the C2M2 format, it gets uploaded, and then we have the search engines that people can use to search across Common Fund programs. So once you upload your C2M2 metadata, you also like some process data like gene set libraries and knowledge graph assertions and attribute tables and highly processed data, as well as your code assets like your APIs, if you have an MCP server that I'm going to talk about, if you have some tools that you developed, and if you have scripts that convert the data to the raw data into that process data, we sort of want to publicize this for you. And once we have all of those assets, we build a bunch of at a higher level, bunch of tools that can be used to query the data across Common Fund programs, including gene set enrichment analysis, search engine, and then some other tools that I'm going to discuss. So this C2M2 model was established in the first phase of the CFDE, and it was really headed by Owen White, who is like also a big figure in this field, and he was in charge of the entire CFDE in the first phase of it, and that's what they came up with. And I was involved in it. I'm also an author on this paper. But in that paper, it's described like how to harmonize data across resources by metadata. And it's a pretty simple idea, and not going to go in too much details of what they established, but it is a metadata model. So that means it has ways to describe fields, and it includes some information about what's in the file. And it is like, it could be a biosample, it can come from a specific assay, it can be including some compound or some a description of genes, and all of those things are potentially there. And it's all produced in a bunch of TSV files, or like just tables that have information about the data. And the files that you guys are potentially getting from the data generation centers, those are potentially can be described using this frictionless standard. And the frictionless is it's just a bundle of spreadsheets, and then there is a metadata model that sits on top of it, and this is called the C2M2 cross-cut metadata model. So one of the only requirement really to interface with the CFDE is to have someone from your team, once you start getting the data, to put in those metadata bundles and then upload them. And the uploading process is very simple. You just get an account, and then you drop the file in our server. So once you have the data uploaded, you're going to get listed here. And this is just, I had to cut because there's 18 programs that are in this table. And then the different types of contribution that they made to the CFDE is highlighted. So if there is a file, there's going to be a link to it. So C2M2 is the main requirement, but also one of the things that we want to add is this process data, like gene set libraries, knowledge graph assertions, APIs, things like that I'm going to talk about. So to do the C2M2 submission, we developed this command line interface that will help you to do it. So it has documentation, and it has different tools. You install it with, it's like a pip install, it's in Python, and then you can run this program to not only to help you construct those files, but also to validate them. So there is like a validator that you run, and then it gives you some output of what you potentially need to fix. And all of this is in documentation on the CFDE Workbench website. One of the things that happen also when you upload, we do this fair assessment with Fair Shake. So Fair Shake is one of the tools that we developed maybe 6-7 years ago. And it is an idea of assessing the fairness of digital objects. So there is also a paper about Fair Shake that was published in Cell System. And the idea is that you create some metrics for fairness. So you can check, for example, if a data set has proper IDs for, let's say, genes. Does it have the IDs that come from some type of source that is established, source for identifiers? So that could be one metric. And then we construct rubrics. So rubrics are simply a collection of metrics. And then we apply this assessment to your data set or your tool or and you can define what the assessment would be. So it's sort of like a self-administrated test, but at the end you get this fair insignia, and every square in the insignia is a metric, and then you get sort of like a score of how fair your data set is. So you want to reach, like, aim to be fair, more and more fair, and the whole point is pointing like what you could do to the data to make it more interoperable and fair. Once all of the metadata is coming together, it all goes into the search engine. So there is this data matrix that I showed you that is just listed data and people can download it. But also we implemented the search engine that you can query across the data set. So I think one of the things that I'm presenting, this is about the CFDE, but also I'm assuming that now you're building also your infrastructure for the complementary, and you also have to deal with this issue of diversity of data sets. So if you could potentially adapt some of the principles or even the code, it's all open source. And then the search engine, it uses Elasticsearch, and right now it has those 3 million files, and all those files are annotated, and then you can find data sets this way. So if, for example, if you look for liver fibrosis, you get data sets, and some of those data sets, they have a DRS URL, and that means that you can get the files, the original data files, and you can pass them into a cloud environment through this DRS, and you can create like a bundle. So you have the shopping cart, you can add a data set into the shopping cart, and then you have this DRS bundle that you can use for taking all of those data sets that you found from the search and bring them into like a cloud compute environment. And this on the left is liver fibrosis, on the right is dexamethasone search. And then you see that they get like a lot of files and other assets, process data sets that have those terms from those different programs. So in this data matrix, the next column, so I talked about C2M2, and you can talk about it maybe later if you have more questions about it. It's sort of like the main thing, it's the C2M2 that everybody is able to comply. But the next thing is the gene set libraries. So one of the things that my lab sort of like push is this idea that you can do a lot of data harmonization by converting everything to gene sets. And if some of the data sets fits that, it would be nice to have gene sets that come from complementary into this ecosystem. And then those gene sets, their format is very simple. It's just a name of the set, maybe a very short description, and then the genes that belong to that set. And this is tab delimited text file that has that information. It's called GMT. And once you have the data in that format, we can add it to this tool that is a part of the Workbench, where people come in with their gene sets, and then they do in a query, and then they find gene sets that match their gene set from across programs. So right now we have about 12 gene sets, 12 gene set libraries created from about 8 programs. And then you get to query all those programs for overlap with your research gene set. One of the things that we also doing with those gene set libraries, we cross them. So you can take gene set libraries from one Common Fund program and gene set libraries from another Common Fund program and just see what are the overlapping genes. And we could find some interesting hypotheses. And in this particular case, we took the GTEx data, we converted into aging signatures. So GTEx is a Common Fund program that profiled using RNA-seq post-mortem tissues from donors. So those are healthy people that sign up to give their body to science. And then when they died, this GTEx consortium took their tissues and did RNA-seq on 54 of their tissues. And then now you have about 800 humans and their gene expression. And those are coming from different ages. So what we could do, we could compare the young people, like the people from 20 to 29 that donated their bodies and had their bodies profiled, to elderly people, find the genes across each tissue that are up or down. So we see that, for example, the blood of like genes that are upregulated in blood as you age are also downregulated in exercising, in the bloods of exercising rats. So the MotoPack program at Stanford, they study the effects of exercise on the body. So they have acute exercise and aerobic exercise, and they're profiling rats and humans. And they're doing adipose, muscle, blood, and in rats, they're doing all of those tissues. So we find a lot of overlapping genes, or significant number of overlapping genes of after 2 weeks of exercise in a rat, in the rats of a female rat blood, and genes about increasing aging. And now there are 35 genes that are overlapping, and those genes are interesting. And then we had this button called GPT hypothesis, where we give the terms and the overlapping genes to ChatGPT to form an hypothesis of why those genes potentially are, why there is such an overlap. And it finds that those genes are involved in blood clotting and also in, cholesterol metabolism and triglycerol hemostasis. And so there is some lipid metabolism and blood clotting genes in that 35 genes that are critical. So this is just an idea of like how we are finding things across Common Fund programs. The next column in this data matrix is knowledge graph assertions. So I'm assuming that all of you probably heard about how a lot of people are now in the last maybe 5 years are excited about knowledge graphs, and how knowledge graph can be used for multi-omics data integration. And we are also part of this CFDE partnership called Data Distillery. And it's an idea of having a knowledge graph for all of those Common Fund programs and other knowledge graphs from other sources all combined, and have this massive knowledge graph that you can query for biomedical discovery. And the people behind this are Jonathan Silverstein from UPIT and Deanne Taylor from CHOP, that are sort of like heading this idea of knowledge graph. So I should give credit to them. for sort of like spearheading this project. And so the idea is taking all those data sets from all of those common fund programs and make a knowledge graph of that. And what we contributed to this is this UI that interacts with a Neo4j database. So knowledge graphs in general, they all get stored in this Neo4j database. It's a company that sells knowledge graph databases, and their first tier of usage is free. So everybody is using it for storing knowledge graphs, but they don't really have a UI. So our team built a UI on top of a Neo4j database using Cytoscape. And we use this UI for this project. And this is an example of what you can do with it. You can enter two terms, and then you can find some connections between terms that are in the knowledge graph. That UI was published, it's like open source, and it was published in current protocols. And it can be, we already use it for a bunch of projects, but also it's something for you to consider if you are taking the complementary data and building knowledge graphs, and you need a UI. This could be a shortcut or something that you can use. One of the things that we used it first was this other CFDE partnership that called ReproTox. So I thought that was very relevant to you, because ReproTox is the idea of like reproductive toxicity, combining data from different common fund programs and other sources for finding genes and drugs that potentially cause and for this we combine data from IDG, from links, from GTEx, and some other databases that connect genes, birth defects, and drugs. So the drugs are increasing or decreasing the expression of those genes. Those genes are associated with those birth defects. And then those drugs also may cause some side effects that could be associated with the birth defect, or the birth defect could be a side effect of the drug. So all of those triangles can be stored in a knowledge graph, and then you can find some novel associations or expected association, things like missing links in those graphs, or connections between a gene and a drug that was not known or was not recognized. For example, if there is a mutation that causes, let's say, abnormality of the limbs, and then a drug is also known to cause women that are pregnant to have that birth defect, then if the drug also increase or decrease the expression of that gene based on links data, then we can find some mechanistic explanation of the connection, the genetic connection. So the genetic connection does not give you mechanism. And now you can sort of like start seeing the mechanism. And then you see that here on the left is the knowledge graph visualization of the database that was stored also in this Neo4j database. So we've been, this is sort of like a step back. In 2014, my lab published this paper called Lean Data Integration in Systems Biology. And we are already thinking about how you can take all of those resources and databases and projects and converting them to networks, which are now called knowledge graphs. You can make gene set libraries, and you can make those bipartite graphs. So you simplify all of those types of data into those abstracted high-level forms that throw away a whole lot of information, but are very useful for data integration. So attribute tables, it's also probably will be very relevant to your project where you want to do machine learning. And so you have some type of like, you have rows of examples like the expression of genes, and then some class that you want to predict, and then those go in attribute tables. I'm not going to talk about that too much because that's sort of like a vision that hasn't been realized yet. But this is what you input into a machine learning framework. And now I'm going to switch to the code assets that Common Fund program contribute to the CFDE. And this is a little bit more abstract and also in the works. So so before going to the code part, a little bit more about the metadata and data submission system. So there is all this documentation that you can look at on the CFDE Workbench documentation portal. And when you click the submit button on the main menu, you go to these collections of documents that are there to explain to you how you can submit and what the submission system looks like. The submission system, because there's only few people from every center that gets permission to even see that system. We need like names and emails of a couple of people, and there are either submitters or approvers. So the submitters are responsible for uploading the files, and then once the submitter uploads the file needs to be approved by the approver. So the approver could be like Gustavo, and you'll get an email, hey, there is a file that you need to approve to have it listed on this data matrix. So that's how it works. So now going to the code assets, and instead of going to the code assets directly, I'm going to tell you about some other feature on the CFD Workbench that potentially is relevant to you, which is this chatbot or AI assistant. And this is agentic AI type of system or search engine that also can be used to interface with this Workbench, and in more like in a free text conversational type of approach. We are using the OpenAI responses library, and we also upload like a bunch of documents. So for example, for every Common Fund program, we upload their landmark paper. So the chatbot knows about the program, not just by searching the web. It also has, the primary source about the program. If somebody is asking information about the program, it interacts with several MCP servers. So MCP is sort of like the newest hot kid on the block that everybody's talking about. And we have MCP servers for the Workbench, for the search engine, but also for some other tools. And also the idea is that the Common Fund programs will have their own MCP servers, and there's a way to make the chatbot aware of different MCPs. So there is a list of them that the chatbot talks to. I will talk about it more. And then besides answering in text, it also can make tables and figures. And it really, the engine behind it is this playbook workflow builder that I'm going to elaborate on. And the most important thing is that the NIH does not want it to hallucinate. So it has to be very much guarded, and it can only do very limited things because we only want to make sure that it does not do anything that is wrong. And we'll go over that a little bit more. So this is some examples of how this chatbot works. So for example, you can ask it, what is the expression of AKT across tissues? And it produces, it queried the GTEx API and produces a plot of all of the average expression of AKT across all those tissues. You can ask it to about a specific glycan, and then it knows about the data from this other Common Fund program that's called Glygen, and then it queries their API and then find information about the glycan. And this is like a long summary of all of the information from their database. And then we also can ask it, like, how do I combine data sets from these two programs, SendNet and A2CPS? And then it gives you some ideas of how to combine data by simply reading their landmark papers. And right now, the ChatGPT knows how to figure it out. So it gives some suggestions of how you can do it. So a little bit more about the playbook and how we are dealing with workflows and handling, the processing of data and the producing data analysis. And this started in 2016. We sort of like were one of the first people that jumped on the Jupyter Notebooks bandwagon. And then we published this paper in this new journal at this time. And the idea was that you can create an article that also has code in it. And then you also give that article to the community, so you can do RNA-seq analysis with that code and this paper for some other stuff. So this was analysis of Zika. So Zika at the time was the virus of interest. And then we created an RNA-seq analysis workflow for some published Zika dataset, and then made that available in the Jupyter Notebook, and then published that as a paper in this journal. And then sort of like the next step from that, so that was a good idea. It was kind of successful and exciting. So the next thing is that we realized that you can actually execute those Jupyter Notebooks dynamically. So you can give it data, execute a notebook, and then just present the executed notebook as a report for people. So we stuck with RNA-seq, and in this application called BioJupies that was published in 2019, I think. There's no citation. I was looking for the, I should have put in also the citation, but it was published in, I think also in Cell Systems or something, but it's an app that is still being used by the community to align your FASTQ files and do RNA-seq analysis. And it is working a little bit like TurboTax, so you come here and you click on Get Started, and then it shows you some, I guess in the next slide, it shows you, do you want, you have an expression table already, or you want to start with your raw FASTQ files. And one of the nice things is that we also offer still free sequence alignment. So a lot of people, you have to go to a data center or you talk to bioinformaticians. So a lot of biologists really like it because they can just upload their FASTQ files and get their FASTQ files aligned. But also you can come in with an expression table. And then once you have your data, you have to make some selections. You have to click your control and perturbation samples. And then you have a buffet of tools that you can use. So you select from the buffet of tools. And then once you click submit, you get a report, and it has all of the analysis of all those tools. And one of the things that made it popular was this blog post by Lior Pachter. I don't know if you guys know about this blog of Lior, but there was a lot of controversy, and people were asking me, did you get scared when you saw that Lior made a blog post about your work? And I said no, I didn't do anything bad. I was not scared, but that because it's so popular, it sort of like increased access. And the real thing that I want to just say is that it sort of like creates this concept of a paper in 4 minutes. So we sort of like carry that concept of like writing a paper in 4 minutes or in one click of a button. And so the next thing was like, okay, this is very specific to RNA-seq, but now we can start taking that concept and extend it and apply it to any type of bioinformatics workflow. And this was the idea of Daniel Clark to create those appeters. So I came up with the name appeters, but Daniel came up with this idea of like, oh, we can use Jinja, blah, blah, and then we can convert those Jupyter notebooks into apps. So there is this SDK that Daniel wrote that takes a Jupyter notebook with some templates that are defined in the notebook and convert it to a full stack Dockerized web app where you could put in your data, like it's an input form, like an HTML input form, and then you upload your data and you press submit, and then it sends the data to the notebook. It executes the notebook and give you the executed notebook as the output. And we have already like over 100 of those appeters that either we developed or the community developed, and they're all served on this appeter catalog. So those are lightweight bioinformatics applications that are generated from a Jupyter notebook. So if you have a Jupyter notebook written in Python, hopefully you can add some very simple code to the global variables or the inputs of the notebook, and then you compile it with our SDK, and then you get an app so that you can host. So this is appeters. But I'm getting there just to talk about this playbook, which is the newest iteration of this concept of getting workflows into more useful, in like automating the bioinformatics process. And the playbook workflow builder was also it was a CFDE partnership. And the idea is that we have APIs, and all of those APIs can potentially talk to each other, but you need to encapsulate it. So this was pre-MCP world, but it's the user interface has those cards, and those cards, you just need to click on them, and then you build a workflow visually by clicking those cards. So the first set of cards are input cards. So those are things that you want to start with. Like, for example, I want to start with a table, count matrix of expression, or I want to start with a gene. And then once you click on that card, then you see, okay, what I can do with that gene. I can potentially get information from GTEx, or I can get information from links, and then I can start expanding that card, and then build a workflow, and then all of those workflows are connected into this knowledge discovery graph. So this is not so it's more of a collection of APIs, connected APIs, but also it has input and output nodes. So this is, it also has a chatbot that builds the workflows, tries to build the workflows dynamically, and then it creates those reports. So this is a workflow that does analysis of study from the GeneSet Omnibus. Like this is just a typical RNA-seq study that the workflow takes in. It just needs the GSC ID, and then it the chatbot knows how to build a workflow or find the right workflow for that input. So this is an example of a workflow, and I thought I put in. So the output of that workflow is a paper, and this happens automatically. So it writes the abstract, it does analysis, and it has the citations, and the citations are verified citations. So we have a way to do a PubMed search, and then let the PubMed search summarize the text, and then making sure that the citation is correct, because it is coming through the abstract and the PubMed API. So I sort of like skipped this part, but what I wanted to say about the workflow builder is that so each circle here, or each node in this network, it's called a meta node, and it has defined inputs and outputs. but it can be of three types. So it can be an input, that's something that accepts data. It can be some type of transforming meta node, that it does something to the data. It's like convert a gene into a table of expression across tissues, and that will be using some API, or it's an output node, and that's produce some visualization, or like a bar graph or a table or some other. So those are the three types of nodes that you can have in this network, and then you build those workflows from that. So this was the example that I explained, and this is the output of that example. It's a PDF. You can also get it in the HTML format. The PDF is also a meta node. So make a PDF of this report is a meta node in the playbook workflow. And that takes a latex bundle that was generated from the report and then compiles it into a PDF. So we could talk about maybe after the thing about the details of how this is implemented. And then we started, thinking about, okay, we can now do this idea of making papers generated automatically for other stuff. So this is an example of creating writing a paper automatically by combining data sets from GTEx, MonoPack, and Metabolomics Workbench. So this is actually 4 sources of gene sets. So also include Rama Geo, which is the automatic way to compute gene expression signatures from Geo. And then by crossing all of those gene set libraries, we find some really interesting crossing of some potentially here, like we found this a particular drug that can potentially be a modulator of aging, exercise, and metabolism. And so it writes the abstract, and it produces a Venn diagram of all of the gene sets from the 4 sources. And then it talks about the genes, and then it does enrichment analysis on those genes, and then it interprets the enrichment analysis results. And so you have a paper. So I think the last project I want to talk about is this liver portal, which is a collaboration between the CFDE DRC and the CFDE Knowledge Center. And this liver portal, I think, is also potentially relevant to you because the liver is a major organ for assessing toxicity, dealing with toxins. But this project tried to combine all of the Common Fund data sets around the liver and create a liver resource for the community. So that's the idea. And this is being developed at the Broad by Jason Flanick and Noel Burt. And they also have somebody from UC San Diego that is a part of their center, and Shankar Subramanians from UCSD, who's sort of like is person that came up with this idea of focusing on the liver and doing some project specifically for the liver. So the way it is divided, there's some people that are doing like single cell data wrangling. There's some people doing like the integrating data sets from different Common Fund programs. And what my group is doing is like we have this liver disease focused effort. And we have those 14 different liver diseases that we are producing articles and analysis based on those diseases. So the way it works, it finds genes that are lowly studied in the disease, but we think that are relevant to the disease using 2 methods. And then also it does differential expression analysis from published studies of the disease versus normal. And then it produces the expressed genes and also look at those genes that are predicted to be a part of the disease. And then it writes those articles about each of those diseases based on that analysis. And then it creates this graphical abstract, and then it produces some figures and figure legends. and this is still in the works, but it is sort of like a paper about each disease. So just to acknowledge the contributors of this, so this is a collaboration between my lab and Shankar's lab, and this is the grant for the CFDE Workbench, is the OT grant. And yeah, I think we can take some questions and have a discussion about it.